So like I was saying, here you can see the ectodermal competence and the ability to respond to the optic vesicle inducer in Xenopus. Xenopus is an amphibian, it's a frog. So here the PAC6 is acting as a competence factor. So the cells must be having that ability to respond to the inductive signals, and that ability is known as competence, like I said. And also there must be something which is responsible for producing this ability inside the cell, which is known as competence factor. So as you can see, this is the head portion and this is the trunk portion of Xenopus. So in case of normal induction, the lens is formed wherever the optic vesicle is present under the ectoderm. So ectoderm is made competent by the presence of optic vesicle under it. So optic vesicle is the inducer which is responsible for this PAC6 competence factor, and thereby it is making the ectoderm competent to develop into the lens. When the optic vesicle is present under the trunk ectoderm, as you can see, lens cannot be formed because this ectoderm, it is not competent. It cannot induce this ectoderm to form the lens because there is no PAC6. And in case where the optic vesicle is removed, then you can see the head ectoderm fails to form a lens. Also, when other tissue is present except optic vesicle, then also there is no induction. So that means it is clear that the optic vesicle is responsible for the inducing signal, and also the ectodermal competence should be there, which is given by PAC6. The PAC6 as competence factor should be present for the formation of lens by optic vesicle. And next is another term which is known as lateral inhibition. Lateral inhibition is a process of cell, like how the cell is controlling the pattern of differentiation of its neighboring cells or adjacent cells. So here you can see this blue colored cell is a differentiated cell and the white cells are all undifferentiated cells. So either it can go this way or it can go this way. So here in this way, as you can see, it is inducing all its adjacent cells to become differentiated into the same fate or become one of its own kind. Like here all the cells have turned blue. That means it is known as lateral induction, means side, lateral means side, and induction means obviously it is inducing the cells to move on to the same pathway and follow the same fate. If what happens, if just the reverse case happens, like it is inhibiting the cells which are adjacent to it to become something like its own, like to follow the same fate as its own, but to follow an alternative fate or to follow some secondary pathway, but not the pathway which it has followed. So here you can see that the cells have turned into something else, not the blue, not the white, but a striped one, which indicates it is a third kind of cell. And why is this forming? Because it is being inhibited by the differentiated cell in the center. Next row, you can see that there are certain cells which are differentiated. So here in this way, it is creating a regular pattern or a regularly spaced pattern, and so that all the cells are not differentiated at a particular localized area, but there is a spacing between them. There is a pattern of differentiated cells in between undifferentiated cells or in between alternately differentiated cells. So this process is known as lateral induction. An example is the delta notch signaling pathway. Delta is the ligand and notch is the receptor. So what happens is that the delta activates the latter in neighboring cells and direct them in alternative pathways. It does not allow the neighboring cells to follow the same pathway as it has followed, like the cell itself has followed. Next is the fate map. Fate map is simply a diagrammatic representation of the prospective fate of each cell or each part of an embryo. So you can trace it back to the final developmental stage, and you can then say that okay, this portion, the cells of this portion will form the epidermis. So here a map is drawn, and according to the regions of the cell or where they will differentiate into, that is named accordingly. So it is traced, the cells are traced, and their lineage shows that these cells will form the epidermis, these will form the neural plate, and in the interior, if you see the interior side of the blastula, you will see that this portion will form the heart, this is the mesoderm. So in this way, the different portions of the embryo are labeled according to the part or the body, according to the region it will produce in the final development, in the course of development. So it is very helpful because in this way you know that exactly which cells of the embryo form what in the later stages, and it helps to understand how the cell movements have taken place during gastrulation, how the ectoderm, mesoderm, endoderm are getting arranged, and exactly what is getting formed from mesoderm, what from ectoderm, and what from endoderm, and so on. So how is this? Basically, this mapping is done by mainly three methods. One is the vital staining. These are nothing but certain dyes called vital dyes, like methylene blue, neutral red, Bismarck brown. So these do not interfere in the normal pathways or normal activities of the cells, but the dye remains, or the dye is contained within the cell till a long time. So you can trace it, like wherever is it going, in which body part it is going into. So you can now understand, okay, I stained this portion of the embryo with this particular dye, and this color is being shown in this body part of the embryo. So that means if it is a muscle, then you know from which cells or from which part of the embryo muscle gets formed. Like that, you can also use carbon particle marking, like the cells can be coated with a carbon particle, and that can be tracked, and then you can know that which pathway is it following. Also, you can use the radioactive isotope labeling with C14 and P42 with radiation. You can easily understand which cells are exactly forming what. So what is cell lineage? Cell lineage is basically the history of a cell, like from what was its origin and what are the stages it went through, and what is the final stage or what is the final fate it has become. So this lineage is like, from here you can see that it was a stem cell, and from here different germ layers have formed, from different germ layers different subclasses of stem cells have formed, and then different progenitor cells, and from there the final cells like astrocytes, neurons, hepatocytes. These are terminally committed cells. They will not form anything else. So this entire pathway, like from embryonic stem cell to endoderm to endodermal progenitor to a hepatocyte, this is the lineage of a hepatocyte. The history starting from its birth to its ultimate fate. So some organisms like C. elegans have invariant lineage, which means they have all the organisms of the same species have the same, almost the same number of cells. Around 1,000 somatic cells are present in each C. elegans. So it is very easy to study developmental biology in C. elegans because of their first transparent body. You can trace each and every cell of the body, and because of their constant number of cells. The cell number does not change. But in case of humans or any vertebrate, you can say that whatever cell number I have in my body is not equal to whatever cell number you have in your body. Although we are of the same species and everything, but still you cannot say. Why? Because it depends on the number of cell divisions, the number of cell differentiations that has taken place inside my body and that has taken place in your body. So that will always be different. So we have variant lineages, but organisms like C. elegans have invariant lineage. The pattern of lineage and everything, the number of cells and everything will always be same. Number of cell divisions, differentiations will always be same in each C. elegans organism. Next is the stem cell. So what is a stem cell? Like I've already discussed while doing the potency, that stem cell is something which has the potential to give rise to a lot of cell types. Like it is an undifferentiated cell, and that can divide either to form a cell of its own kind, like it can give rise to more stem cells. This is known as self-renewal. Or it can give rise to a number of specialized cells or differentiated cells. This process is known as differentiation. So stem cells can be of two types. based on their source. Like what is the source? It is either taken out from embryo, then it is called an embryonic stem cell, or it is taken out from adult, then it is known as adult stem cell. So in case of embryonic stem cells, it is mainly located in the inner mass of blastocyst, inner cell mass of blastocyst. And in case of adult, it is distributed tissues throughout the body. For example, bone marrow, we have the hematopoietic stem cells. From there the blood cells give rise to the blood cells. So embryonic stem cells, like I already told you during potency, when I was doing potency, that this is pluripotent, but adult stem cells are multipotent. So the potency has decreased. The cell fates are restricted much more in case of adult stem cells. So you can understand that they are multipotent. And what is the role? They become, this inner cell mass can become any cell of the embryo, any cell type. Therefore, they are pluripotent. And this adult stem cell are essential mainly for repair, like when there is a cell deficiency, then it produces the more number of cells, and therefore adult stem cells are very important in that. But they are tissue specific, like only it will, like example bone marrow, it will only form cells inside the bone marrow. They will not form any other cells, or they do not have the ability to form any cell of the adult body. Therefore they are multipotent, not pluripotent. So based on their potency, you already know that stem cells can be divided into totipotent, pluripotent, multipotent, oligopotent and unipotent. I am not getting into details because I have already discussed this. And another term that is very important in this differentiation is the community effect. So what is the community effect? Here you can see that there is only one muscle precursor cell. That means it will ultimately differentiate into a muscle cell. But it is not forming a muscle cell. It is forming something else. These are other differentiated cells. Gray color. Alternatively, when you see that there are a lot of muscle precursor cells, and these are white cells are some other cells, but after differentiation all these precursor cells have become muscle cells, but these other embryonic cells have become some other differentiated cells. So what is the difference between these two pictures? The first one is that there is only a single cell. So here the other neighboring cells are inducing an alternative signaling to this particular precursor cell, and it is inhibiting that it cannot form the muscle cell to which it is supposed to form. Instead it is getting differentiated into something else. But here, as you can see, since there is a group of cells, group of precursor cells, so they are able to maintain their pathway. They are able to maintain whichever fate they are supposed to follow, and they are forming muscle cells accordingly. So what can we learn from here? That when there is a group of precursor cells, they can maintain their expression and differentiate in a coordinated manner, and it can change the collective cell fate. But when it is present in a single number, then it is not able to do so. Why? Because the inducer of this particular precursor cell is very less compared to the inducer molecules of these other embryonic cells. Whereas here the concentration of inducer molecules is very much higher because all these cells are together secreting the inducer molecules. So this we can also call it as autocrine signaling. In autocrine signaling, I already told you that the inducer and responder is the same cell. So here the cells are producing the inductive signals and getting induced also by it. So they are responding to it as well, and therefore they are controlling their own differentiation and controlling their own cell fate. So this is known as community effect. It is the effect of the community because of which the cells are able to change their collective cell fate. This will not happen when it is present in a single number and not in a community. So next is genomic equivalence. Genomic equivalence is whatever the DNA content of a particular cell is, that DNA content is conserved throughout the entire organism. Like the organism will have equivalent genetic information in all its cells. because the DNA is same in all the cells of a particular organism. So there are a number of ways that it has been proved, and now we already know it is a well established fact that DNA is conserved and in all the cells of a particular body. But only in some exceptional cases there is some loss of DNA, or there is some gain of DNA, or there might be some rearrangement. But other than that, the most cells, they exhibit this genomic equivalence. They have the same equivalent genomic content in all the cells. So here if you see that this is an unfertilized frog egg. The nucleus is destroyed, and from an adult frog the skin cells are cultured, and the nucleus from the skin cells is injected into the unfertilized egg. So what will happen is that this nucleus contains the genetic information of a frog. Now there will be a normal blastula formation and a tadpole will be formed normally. Like whatever is the developmental pathway of a frog, it will follow that only. So you can see that a skin cell nucleus can be replace a normal unfertilized egg nucleus. Why? Because all the nucleus present in a frog body will have the same genetic information. It will eventually give rise to a frog only. So from here, this nuclear transplantation method also, you can see why genomic equivalence was proved. Next is imprinting. So imprinting is basically a type of genetic regulation where one of the two genes, like every gene is present in two sets, one coming from the paternal parent and one coming from the maternal side. So depending on the gender of the parent from whom the gene is passed on, one set is getting expressed and one set, or the reciprocal one, is getting repressed. So imprinting is nothing but inactivation of one set of chromosome coming from one particular parent. So there are some genes which show imprinting. Like here, if there is an example of body color given. So what happens if the gene is maternally imprinted? You can see that the father is black and the mother is white, and their offsprings are black. Why? Because the maternal gene is imprinted or inactivated, and only the paternal gene is being expressed. What if the body color gene is paternally imprinted? Then what will happen? The maternal expression will be there. So if the mother is white, then the offsprings will also be white, whether it is male or female. And the paternal gene showing black color will be imprinted or inactivated. So in case of imprinting, the chance of diseases or always increase, because there is no such no other complement of the gene is repressed or inactivated. Therefore we know that already there are certain diseases which are in heterozygous condition, which is in a dormant state, but only in case of a homozygous state, if the gene is in homozygous condition, then the disease is manifested. So in those cases, when there is no such dominant allele present, if the dominant allele is a wild type allele and not a mutant allele, then if it is inactivated, then that disease will obviously be manifested, because there is no heterozygous formation, or there is no other complement of the gene to balance it. Therefore, imprinting is a very important cause of genetic diseases. Next is mutants. So what are mutants? Whenever there is a mutation, that is a change in the normal nucleotide sequence of the organism, then there will be a mutation. So mutation is basically a very rare incident, but still it has a lot of repercussions, but also it can lead to better chances of survival, and it can also be advantageous. So this is an example of a mutant. Like you can see that normal wild type tigers have, we already know, they have yellow fur with black stripes, and when it's a mutant, it's a white fur with black stripes or without stripes also. So there can be several types of mutants, and its population will always be lower than the wild type population, but still it is there. So mutation can be caused by anything, it can be by ionizing radiations, or it can be by mutagens, certain chemicals, or it can be by viral infections, or it also can be because of certain wrong DNA copying or DNA complementarity during cell division. So all these chances are there. And the last topic that you have in this portion is the transgenic. So transgenic in analysis of development. What is a transgenic? When a foreign DNA is introduced into an organism, like when one or more DNA sequences from another species is combined together and introduced into an organism by artificial means, then that is known as transgenic organism, and the process is known as transgenesis. So usually a small segment of DNA can be injected into an embryo or a fertilized egg, and then a transgenic animal can be formed, or in case of plants, any portion of the tissue can be introduced into a plant, and therefore a transgenic plant can develop. Such can be the methods. So here two methods are explained. One is where embryonic stem cells are, the DNA is injected into the embryonic stem cells, and from here the cells are expressing these cells are expressing the desired gene, because the DNA you are incorporating into these cells. And when this DNA is injected into the inner cell mass of the blastocyst, then this blastocyst has the DNA information of the foreign DNA that you are introducing, as well as the DNA of the embryo already, which is the from which the blastocyst is taken. And if this blastocyst is now implanted in the uterus of a foster mother, like this is a mice experiment going on, so here the mother will bear this embryo, and it will give rise to an offspring. Now you have to test whether the offspring is containing the foreign DNA or not, whether the transgenesis has worked or not. By how? By if you get heterozygous offspring, then you will be sure of that. And if you want only the foreign gene to be expressed, then you have to mate the heterozygous offspring to create a homozygous transgenic strain. Same way you can also do by injecting the DNA into the fertilized egg. Like these are cultured stem cells, and here you are introducing the DNA, but you also can incorporate the DNA in the pronucleus of a fertilized egg, and then you can directly implant this fertilized egg in the foster mother, and then the rest of the process is same. So instead of a blastocyst, you can inject into a fertilized egg. So these are the two methods. There are other several methods, and I'm not going into details because it is not you do not require this in details that much. And the use of transgenic animals is very important for the study of dev-bio, because you can understand that you can create genome which has certain the genomic complement of diseases, and then you can study those diseases in the mice which show those particular strain. And also you can study the importance or the effect of different drugs in the in what are the different effect of drugs in those transgenic mice, and there are other functions of transgenics in this developmental biology studies. So with this, I have completed the basic concepts of development, which is the first portion of the unit 5 dev bio portion. So in the next video, I will move on with gametogenesis. Thank you.